Welcome to the Lean Blog Podcast. Visit our website at www.leanblog.org. Now, here's your host, Mark Graban.
Hi, this is Mark Graban. This is episode 89 of the Lean Blog Podcast for April 20th, 2010. Our guest today is Keith Russell. He is a global continuous improvement leader at AstraZeneca R&D. I met Keith at a recent Lean and Six Sigma conference for the pharma R&D industry where we both presented. Keith has substantial in-house R&D experience across the entire value streams and currently works in global drug development. He is particularly interested in design and innovation and the application of systems thinking to complex business problems. He has a Ph.D. in synthetic organic chemistry from the University of Cambridge. So in this podcast, we discuss the applications of Lean and Six Sigma in a space we've never talked about here, pharmaceutical development. So how do you use Lean principles in such a technical field, and how do you engage the highly educated scientists who do this work? I want to thank you for listening. You can listen to previous episodes by going to leanpodcast.org, and you can visit me on the blog at leanblog.org. As always, thanks for listening. Thanks for joining us, Keith.
You're very welcome. Thanks for inviting me, Mark.
So I was wondering if you could start, as we do with a lot of guests, introduce yourself and your background, and particularly how you got first introduced to the Lean methodology.
Yeah, well, it's probably useful just to kind of go way back just to give you my roots, which were in chemistry. So I'm basically a scientist. And I started with AstraZeneca as a research chemist about over 20 years ago now. Mm-hmm. So basically, my role there was to discover new molecules, chemical compounds, hopefully with potential for being drugs at some point, in cardiovascular or CNS or cancer, that kind of thing. So, my passion there was for chemistry, and particularly designing molecules. That's what I did. So, I basically, kind of did that for 20 years in different ways, and moved into the kind of research project leader area, where I kind of led projects across our research area, all the way up to where we basically are giving clinical candidates, if you like, over to our development folk. So, I basically then I've kind of looked at this myself and thought, I had a passion for chemistry, but it was about design of chemistry. I then really had a passion for kind of the broader design and innovation of kind of on the program scale, if you like. Mm-hmm. And actually, with hindsight, it was a kind of a natural step in a way to take that passion for design improvement to even bigger scale, which is kind of, how do we do R&D more efficiently, more effectively, more innovatively? And although it wasn't planned this way, I basically ended up, as I am now, really not doing chemistry anymore, but looking at kind of the bigger problem of how do we do R&D in an improved way.
So with that interest in improving that R&D process, I mean, what was your first exposure to Lean? Was this something that you were seeking out and stumbled across, or something that somebody introduced to you? I'm just always curious with somebody in sort of a non-traditional Lean industry, how you got introduced to it.
Well, I can honestly say that in 2005, beginning of 2005, which is when we started to get into this area, I had never heard of Lean and never heard of Six Sigma. But I was given a problem at that point. I was a member of the research area leadership team. I was heading up the chemistry group at that time. And I was given this problem, which is we need to figure out how we improve the efficiency and effectiveness of the last stage in our research. It's called lead optimization. And so basically I had that problem, and as we did at the time, often we turned to look for technology solutions. Often, we look for what are the biggest problems, where are we failing most. But what happened, which was somewhat of a chance event, was I had a person in doing training for my chemists in design of experiments. And he and one of his colleagues in England had been basically experimenting with Lean Sigma, which was very new. It just started in our company, in some very small controlled areas in the labs. And I basically told him, I was chatting to this trainer, he was a statistician, and I told him what my problem was. He basically ran through what Lean Sigma was, and basically it was essentially that's for me is where it started. Mm-hmm. It was, the problem meets the potential solution, and I was excited from the start because I could see the opportunity right there.
Yeah.
It's had quite an evolution since then, but for me that's where it started, and basically we then took that and applied it to a complex cross-functional project, and a lot of it is spread from that point.
No, I'm really interested in a couple aspects of this. One would be just the general applicability to Lean and Six Sigma in pharma industry, because it is so different than discrete manufacturing, the building of cars, the building of computers. And, in any industry, any setting, people will raise the flag of, we're different.
Yes.
And so I could see one level of that on the pharma production side, the very last stage. And maybe especially so with the scientists and with the development. So, when you talk about efficiency and effectiveness, I'm wondering, or curious about how people react to those words. Is efficiency a scary word, or how do you measure? I guess that's one of the base questions. How do you measure that efficiency, or effectiveness? What are you looking for?
Yeah, the fundamental question about how do we take what has been learned in the Lean area and Six Sigma. We call it Lean Sigma, which is, as you know, the blend of Lean and some of the Six Sigma approaches. How do you take that and extract something useful to help R&D? Mm-hmm. And I think, I've heard you say this. What would have been a disaster is if we'd taken, if you like, off-the-shelf Lean, off-the-shelf Six Sigma as applied to manufacturing and just applied it. Right. And we did some of that. We made some of those mistakes. And just as you're kind of hinting, the scientists did not like that at all. Mm-hmm. Because the scientists are fiercely independent, they're very highly educated and trained, they've got a lot of experience and learning, which tells them that something that somehow they perceive as constraining creativity, and they did, is not good. Something that speeds things up without worrying about, what is the quality or reduces cost, scares them. Sure. So there was certainly a challenge there right from the get-go with some of that, absolutely.
So what were some of the approaches for addressing those concerns? Because, like you said, they're fiercely independent, highly skilled professionals. If they wanted to derail the effort, I'm sure they certainly could. What were some of the strategies or approaches that you took? Well,
in some of this, we've obviously learned as we've been doing it since 2005, but we recognized pretty early on that, unlike manufacturing where you're making the same, or ideally you're making the same product every time, and you really want to minimize variation, you want to make it as cheap, as fast as possible, get that product to the customer, that obviously, is a value to them. The whole concept of value stream makes so much sense when you think in that. But what does it mean for R&D? Because if you think what R&D does, it essentially is creating new value stream opportunities for manufacturing. It's not manufacturing anything. It doesn't make products. It makes opportunities for products. So we had to get our heads around that. So basically, what does value mean in R&D? I think was difficult, particularly as you start to kind of zoom out and think of the bigger picture. So once you start to realize that, what we're doing is applying science. So science is advancing knowledge, and we're using management principles to deliver results. You start to, get your head around this and realize that what R&D is doing is essentially making or generating new, useful knowledge. It's a learning process. And I think that's where the difference is. So you can't know the answer at the beginning. You have to do the work to get the answer. Mm-hmm. And I think that is, the more we appreciated that and the more we realized there are areas of R&D that are like manufacturing and we can apply very similar, very familiar lean approaches, whereas there are other areas that are more in the kind of creative end where we have to use some different approaches. Once we realized that, I think folks started to really open up and appreciate the value of this.
Okay. Now, how long would you say AstraZeneca or in the labs, how long have you been working at figuring out how to apply Lean Sigma?
Well, really since 2005 is where we started in research. And we basically a couple of years of development in across the discovery, the research end, and then it spread into our development area, where we're doing clinical trials, where we're doing our interactions with the regulatory authorities, our process development work, and it's basically spread from there.
Right. So, being a few years into it. Are you able to share either some broad measures of how you would gauge the impact that Lean Sigma has had, or any specific success stories where it led to a real noteworthy improvement?
Yeah, let me pick I got a couple of just real simple examples. A couple from the research end, just to illustrate how you can actually apply some very simple lean principles, which I think is what we're doing here. So for example, in the lead optimization part of research, the chemists make molecules, make compounds, and they make lots of them, and ultimately we're trying to find one that may be a clinical candidate and ultimately a drug one day. But most of the molecules we make are just answering specific questions. So one of the things we realized by doing essentially a big process map with a lot of folks working on this process is, if you make too little compound, you can only answer a few questions. You can only do the first biological test, or the second. If you make too much, and so you have to, if the compound turns out to be good, you have to make more. So you create a lot of rework if you don't make enough. If you make too much, well, most compounds don't make it as drugs. They die. They're not good enough. So if you make too much, you have a lot of waste. So we realized there's actually an optimal point of how much you make that allows us to drive the process through to an appropriate point without being too wasteful, and actually gaining momentum, if you like, and through the project. So that was one of the things we did. That and a number of things, just simple things like that, led to us taking cycle times for this lead optimization phase down from 12 to 18 months as it was, down to about 5 to 6 months.
Okay.
That's one example. There's many other elements that led to that. But there were very simple things like that because we never really looked at it that way before. So lots of low-hanging fruit in a sense.
Yeah.
An example more on the development end, but again, very simple kind of lean principles, is if you look at some of the early clinical trials with volunteers, where we're basically, for the first time, introducing some of our potential drugs into people, and we want to just make sure in a very carefully controlled way, do the molecule, are they tolerated well, and things like that. Well, the way those trials are run is they're run with cohorts of volunteers. It can be 6 to 9 cohorts, and you run them in a linear way, 1 cohort, 2, etc. So we realized that the cohort to cohort cycle time, there was a lot of waste in there because there's a lot of time moving samples around. There's a lot of time getting the analysis done and getting the information back and integrating that information. So a number of pieces of work basically addressing just simple lean wastes, overproduction, waiting time, things like that. We tackle those. That obviously multiplies up now because you've got, up to 9 times you're doing that. And again, we saw something like 50% improvement in cycle times and things like that. So we've done a number of things like that across research and development, if that's helpful.
Yeah. And I'm curious, just to follow up a little bit on methodology. Were these done in the form of, a week-long Kaizen event in the context of a green belt or a black belt project?
Yeah. So the answer is both. So we've used kind of in a way a blend between what you would typically call a black belt project and a Kaizen event, where we found that typically for us the lean approach has certainly been the most fruitful so far. But we have used the DMAKE Six Sigma framework. So many of our activities were in the form of 3-day workshops where, we would map out a process with all the folks that work that process, and basically do what is typical in a Kaizen event.
Mm-hmm.
To, understand, the timing of the steps, understand who's doing what, the handovers. That is the typical approach. In other cases, we've done a very, off-the-shelf, black belt, if you like, DMake project.
Mm-hmm.
So we've used whatever works depending on the degree of difficulty, really, of the problem.
Okay. And, I'm curious to delve a little bit into the idea of, you have a company full of scientists,
mm-hmm.
and in a lot of settings, there's discussion about how, the true essence of Lean or the Toyota production system is the scientific method applied to process improvement, going back to Dr. Deming and to other
mm-hmm.
roots. And I've heard, some organizations say, about manufacturing and healthcare, our goal is to have everybody to be a process improvement scientist, to have everybody involved in this every day. So I'm wondering how, the opportunity or the progress in trying to get people who clearly have scientific training and scientific mindset around things like molecules, which are squarely out of my realm. But to apply a similar mindset to things like process that work in conjunction with the scientific discovery. I mean, what are you finding in terms of getting people to embrace that?
We even before we did the Lean Sigma work, we recognized there was a cycle, and we called it the design, make, test cycle.
Mm-hmm.
That's what we called it, which was the learning cycle, which is akin, to the plan to review or DMake, whichever form you like.
Right.
But basically, for us though, and this perhaps is the difference, it was initiated with a question, right, or a set of questions. And we were looking for answers, which is what the learning was, of course.
Yeah.
When we first mapped out that LO process, this really hit us. You could see the cycles very clearly. And I don't think they've been that clear to folks before, how things iterated. And those cycles continue into development. They just get longer, because now we're running clinical trials to do, if you like, the test part, if you like.
Mm-hmm.
But absolutely. But I think where we can go, though, is to be much more disciplined in the application of that kind of thinking, even now. So, which basically means doing better science, and we're not going to get a lot of argument with that.
So Lean for better science, and then Lean Sigma not stifling innovation, which is probably the other concern. And maybe we can wrap up with some thoughts on that, because you hear this in other industries that are very technology driven. With the Lean startups movement, as people refer to it, or even diversified companies like 3M, where there's a fear or knock that this idea of process will kill or stifle innovation. So, what are your thoughts on that?
Yeah, and I fully understand
mm-hmm.
where this dialogue's coming from, because at least superficially, if you look at Six Sigma and Lean, he's trying to eliminate variation. That's part of what it's about. Because the customer feels that. Okay. Whereas innovation, of course, is trying to create variation, essentially. So that seems like a conflict.
Right.
But I guess you got to frame the problem the right way, I guess. So I don't think the problem is a win-lose problem. In fact, I think we need both Six Sigma, Lean, and innovation, and they actually can work together very well.
Mm-hmm.
So for example, in the design space, in innovative design space, which is obviously a key part of what we do, we have to create opportunities. We have to create ideas, if you like. I should define how I'm thinking of innovation. Innovation, I guess I'd see. I saw this somewhere, that innovation is the transformation of ideas into value.
Mm-hmm. I
really like that.
Yeah.
And if you think about that the entire process, then there is a lot of opportunities for some Lean and Six Sigma thinking, and there's also a lot of opportunities for that creative moment where you generate the opportunities. Six Sigma, it's interesting, that talks about defects, right?
Right.
And defects per million opportunities, is obviously one of the measures they use. Well, we're kind of focusing on innovation on the opportunities, creating those opportunities, whether they come from talking and listening to customers, technology, or other sources. So to boil all that down, I guess I see three types of variation. The special cause, common cause, Deming, right? But also, if you like, the creative cause, which is what drives innovation. And that's useful most of the time. There are some areas where you do not want lots of innovation. A manufacturing process, ideally, there's nothing to learn. It does exactly the same thing every time. You really don't want innovation in that process. Outside of the process where you're trying to improve it and learn new things, then that's different. You need some innovation. So I guess that's how I see it.
Yeah. Well, and I think the dirty little secret of manufacturing, which is my original world, and even the last years in healthcare, is that you'd like to think that it was all figured out and it was just a matter of applying it consistently every day. But there's still an awful lot of room for little small cycles of innovation day in and day out, which is that that magic of Kaizen. So I like the way you said it. How maybe an attempt to summarize. It applies in innovation, and Lean Six Sigma applies in R&D, except where it doesn't. Fair enough.
Yeah. Well, essentially, you have to frame the problem to say, when you look into the details of specific processes, the areas where you want creativity and to create variation, and then at some point you want to actually minimize that variation. For example, when you're making decisions.
Sure.
So it's applying in the right context, in the right place, which is true with everything, really.
Yeah. And
it's how you use it.
Yeah, and in any setting, minimizing variation doesn't mean shut off your brain and have zero variation.
Right.
But so these are great questions to be talking through. I'm glad that you and others in the pharma industry are trying to figure out how to make that work. And so it's really interesting to hear about this in what for me is a very unknown setting. So it's interesting to hear about what the themes are, the types of things that you're working on. So I want to thank you, Keith, for sharing those with us today.
You're very welcome, Mark. Thank you very much for the opportunity.
Thanks for listening. This has been the Lean Blog Podcast. For Lean news and commentary updated daily, visit www.leanblog.org. If you have any questions or comments about this podcast, email Mark at leanpodcast@gmail.com.